I'm going to raise some questions in my talk that I don't think we have a lot of answers to, but maybe as Madame Curie said, nothing in life is to be feared, it's to be understood, and we'll talk a little bit about that. Somebody asked the question about the JAK-STAT pathway. So let's start there. It was perfect. Thank you for asking that question. Check is in the mail. Everything in MPNs we think centers around the JAK-STAT pathway. So this pathway is involved in making blood platelets, but it's also involved in inflammation, as Dr. Fleischman was just talking about. And the key components of this are JAK protein, which we all have and we all use to make blood, but it's mutated in many patients with MPNs, the calreticulin protein, and the MPL protein. And now, just to be clear here, we can't see the mouse. That's all right. JAK and MPL interface with each other, and calreticulin interfaces with MPL. So all of the things that cause the diseases all interplay with each other, and it makes sense, right? If you have a lesion in one part of the pathway, having a lesion in another part of the pathway could give you the same result, right? And that's kind of the high-level concept here. But it raises a lot of questions, too. How do you explain that somebody can have myelofibrosis, or they can have ET, or they can have PV, and they can all have a JAK mutation? How does that work, right? How do you explain that there are different phenotypes, all with patients having the relatively same mutation? The same thing is true for outcomes. How do you explain that if you think about, for example, a JAK mutated patient with myelofibrosis, their mortality risk is definitely higher than it is for somebody who has JAK2 mutant ET. Or if we think about clotting, somebody with a JAK2 mutated PV has a much higher incidence of clotting than somebody with a JAK2 mutated MF. How do we explain that? And then another thing is that, as Dr. Fleischman had alluded to, not everybody who has a JAK2 mutation has an MPN. So there's a concept of what's called clonal hematopoiesis. What that means is that there are approximately 10% of the population walking around with mutations of cancer-associated genes who have no diagnosis, not because they haven't been diagnosed, they don't actually have anything that meets criteria for having a disease. This is called clonal hematopoiesis. Some people call it CHIP. And the mutations that occur in CHIP are all listed here. And of course, you'll notice that one of the most common is actually JAK2. So there are people walking around who have JAK2 mutations who have no diagnosis and no disease. So how is that possible, and how do other people have a JAK2 mutation and have a disease? And it's actually even more complicated than that, because in patients who have a JAK2 mutated CHIP, or clonal hematopoiesis, they still actually have an increased risk of developing a blood cancer down the line, and they actually have an increased risk of having a clot, which, as you all know, that is a big problem in the field. So there are some threads that begin to show commonality, but still a lot of questions. And let's make it even more complicated. This is beautiful work that came out last year, and another paper with the same results came out very recently. People can have a JAK2 mutation for decades preceding even a diagnosis of an MPN. So in this particular study, not getting into the nitty-gritty experimental details, using sophisticated single-cell methods and mathematical modeling, this group was able to identify that, for example, in a 34-year-old patient, the patient acquired their JAK2 mutation 25 years prior to the onset of the disease. Or in this one, even more remarkably, a 63-year-old patient acquired their JAK2 mutation 40 years prior to having the diagnosis of the disease. So it's not just a matter of the mutation being there or not, something else matters. Maybe it's inflammation, maybe it's other things. So what are some of those other things? Well, other mutations happen. They don't happen frequently, but they happen. And so if you look at the y-axis, you'll see that JAK2 obviously is the most common mutation, over 60%, calreticulin 20% at least in this series. But there's other mutations that certainly occur at a high frequency. There's one called TET2 you'll see there, which is maybe 15% or so of patients. And we heard about ASXL1 from Dr. Ritchie this morning, so that's another mutation that can occur. But if you look at the smaller inset, these are all mutations that occur in under 3%, maybe under 1%. There's a lot of other mutations that can happen, right? And if you think about patients with the disease, actually not even that. If you think about all patients, all who have a JAK2 mutation, all MPN patients, all people with CHIP who don't have a diagnosis per se, there's a huge spectrum of mutations, right? And so beginning to understand, do these make a difference in people who have a disease or a more advanced disease is a good question. It's an important question, which we'll come to in a second. Complexity also matters. So one of the things supporting what I just said, do the other mutations make a difference? Well, if you look at the number of patients who have one mutation who have ET at 62%, and that drops to in the 50s with myelofibrosis, PMF, primary myelofibrosis. But the number of people who have 5 mutations is 3% or 4 mutations. So there are more mutations with a more advanced phenotype of disease. That's one thing that this data says to us. And we can extrapolate that further by looking at patients who had an MPN that's now progressed into leukemia. It's not important to know the names of the genes. It's really about looking at the color. And so each bar is an individual patient, and you can just, all I want you to appreciate here is look at the number of mutations. Look at all of these mutations occurring within each individual patient. It gets more genetically complex as it goes on. That has to be part of the answer to this question. Now, going beyond that, and don't be afraid, what I want you to pay attention to is just some key concepts. Don't worry about the letters and numbers. But blood production occurs through a series of highly regulated steps. Blood production starts with what are called hematopoietic stem cells. Now, stem cells are fascinating because unlike other types of cells, they can turn into virtually any other type of cell, and then they can also create more of themselves. If I take red blood cells out of my arm, I can't do anything with them. I can't make them into white blood cells. I can't make them into platelets. They are who they are. They are red blood cells. With a stem cell, you could, using the right modifications, turn that stem cell into a platelet or into a red cell or to a white cell. It's a really interesting and complex process, but it is a series of steps, and that's all that the diagram on my right is showing you, is that it's a series of steps that occur. But if you look at each of those series of steps, and you look at patients who have an MPN, are there differences in where, for example, the JAK2 mutation shows its where it lives, in one way to put it. And so each column here in this panel D is a different compartment in a patient's blood stem production. And what the coloring is showing you is how prevalent the JAK mutation is. So closer to yellow is more high, and the darker colors are lower. And if you just appreciate in the columns, there are differences, meaning that the prevalence of the JAK mutation differs not only amongst patients, but actually within the hierarchy of the cells being produced, right? This is another thing that may lead us towards an answer about how you have all of these different disease phenotypes, or how the disease manifests despite people having the same mutations. Now, do the other mutations actually make a change? What's the evidence? One example of this is the TET2 mutation, which outside of JAK mutations is the most common mutation. And what this is a heat map that just shows you readout from cells. So what are the cells programming? What's the programming of the cell? And the only thing I want you to appreciate is that in the MPN patients who have a TET2 mutation on the far right there, it's obviously very distinct from the patients who don't have a TET2 mutation. So it's changing the genetic readout. Interestingly, though, the TET2 mutation per se doesn't seem to have an effect on the disease itself, although it seems to matter whether or not it occurs first. If it occurs first, patients seem to more frequently have ET, whereas if it occurs as a later event after JAK2, they more commonly have a polycythemia effect. One of the genes that does occur and that we know is, that does have an influence on how the disease behaves is something called P53. P53 is the most commonly mutated gene in human cancer, and it does get mutated in MPN patients, particularly as their disease progresses. So the question that our lab has been very interested in is the biology of this and what does that do for patients. And so what we did, and this is hot off the presses, this paper will be coming out very shortly, is that we asked the question, fine, what if we eliminated this gene? There's 2 copies of the gene, just as there is in humans, in mice, or we mutated the gene. And if you look at the spleens, those are in figures D and E. The top is a JAK2 mutated mouse that gets polycythemia. And if you eliminate this master regulatory gene, it's really clear the spleen is humongous. So that does change the disease, but it's not so simple as if just manipulating the gene makes a difference. It turns out that both copies of the gene have to be corrupted. And if you look at the bottom, if we contrast the first 2 versus the last box, where we lose one of the copies of the gene, it doesn't seem to do anything. If you lose 2, it turns into a fulminant leukemia. Those big ugly cells are leukemia cells. So it's not only the gene, it's how many mutations in the gene. Does it affect both copies? It gets more complicated. Now I'll delve into something else just for a few minutes, which Dr. Ritchie kind of covered. Fine, biology is good, it's interesting, a lot of unanswered questions. What is the practical ramification for patients, right? That's a very important thing. And Dr. Ritchie's talk actually set this up perfectly when she talked about somebody gets their sequencing results back and they want to know, what does this mean for me? A very important question. Turns out these mutations do make some difference as far as we know. One example, for example, here is with patients with essential thrombocythemia. Years ago, we used to say they're either high risk or they're low risk, but it turns out it depends on whether or not they have a JAK2 mutation or a calreticulin mutation. And now we have 4 categories. People can be very low risk, or they can be high risk, or low risk, or intermediate risk. It depends on their genetics. So here is where the genetics makes a difference. Another example of that, which Dr. Ritchie talked about, is what is the risk of progression for people with polycythemia or with essential thrombocythemia? And the genetics seem to make a difference. In these cases, at least, the SRSF2 mutation seems to increase the risk of the disease progressing with other factors associated with it. And the same thing is true of ET, same sort of mutations, but also P53 as well, seems to have an implication about whether someone's disease is going to progress. Now, one caveat with all of this that I'm showing you is that these are relatively small numbers of patients that are being studied. We're talking about 200 or so patients from which conclusions are being drawn. And we have to be really careful about how generalizable that is, right? And I go back to what I said earlier. If you think about all the patients who have a JAK2 mutation, some people have no disease, some people have leukemia, and everything in between. And so when we do studies like this, we have to be really careful how we interpret the data. Mutations have an impact on myelofibrosis. Patients with calreticulin mutations seem to have a relatively better prognosis. So there's another example where it makes a difference. And Dr. Ritchie talked about this. What are the implications of some of the mutations? We know that at least these 4 mutations seem to be associated with the disease potentially progressing. But here again, there's a lot of caveats. There's a lot of clinical data, a lot of heterogeneity that needs to be taken into account. And the bigger the number of patients you study, the more likely you're going to get to a model that is similar to what is actually going on in real life, right? That's a statistical argument. One thing that we always have to keep in mind with all of these prognostic tools is that the data changes. So 10 years ago, if we were to sequence somebody for 30 genes when we saw them, that was state of the art. Today, when a patient comes into Sloan Kettering, and I'm sure it's true for other places, they're sequenced for 600 genes. And in a few years, that's going to be like a horse and buggy. It'll be old-fashioned and not really state-of-the-art. It changes. And that's changed what we know is a risk factor. So for example, these are RAS mutations. These are mutations that actually do increase the risk of the disease progressing. They're not in any of our current scoring models because this is relatively new data and the models haven't been updated. So it keeps changing, and that's also why we've got to be careful about how we interpret this data. Do the mutations affect how patients will respond to treatment? The answer seems to actually be yes there. This is a really great study from a number of years ago from MD Anderson that asked the question, do the mutations tell you anything about whether or not a patient is going to have a durable response to Ruxolitinib? And the answer is yes. Mutations in any of these 3 genes in red, or having more mutations in general, numerically, shortens the time for which a patient's going to have a benefit to Ruxolitinib. So it does matter in that context. Same thing is true for transplant. This is at least one study that has suggested that some mutations that we see in transplant could lessen the likelihood that the transplant is successful. This data is, there's 2 other papers in the literature that don't agree with this paper, so I'm not going to go into the details, but this is something that needs more study. Again, we don't have sufficient numbers of patients. And finally, this is the paper that Dr. Ritchie had referenced. This is the Grenfell paper that tried to put everything together and say, fine, forget about what disease the patient actually has. Let's just look at the genetics and find out how that changes things. And this was close to about 1,800 patients, so a good number of patients, but there are still limits to this data. And one of the important limits is that this was really enriched for patients with essential thrombocythemia, and not the majority were not PV or myelofibrosis patients. So that's a caveat. So with all that being said, right, there are important implications. The mutations that occur aside from the JAK mutations matter. Other things matter too, like the chromosomes or the bone marrow fibrosis. But for us to really have a model and have a real understanding of what all of these things mean, we need a larger data set. We need a bigger, more robust data set to understand that. And that's where one project is underway to address that. And this is from the MPN Research Foundation that's leading a project called the Progression Research Network, which is to really try to identify what constitutes progression. Is it a change in mutation? Is it a change in symptoms? Is it a change in blood counts? And as part of that, genetic data will be captured that will maybe give us a better sense of which of these mutations really matter and in what context. It's not, again, a matter of just having a mutation or not. It's a matter of what else is going on. The hope is that this project will capture data from 25,000 MPN patients. That is quite a bit more than anything that I've shown you, which is all in the hundreds, except for the Grenfell paper, that was 1,800 patients. That's the largest series to date. But the aim here is 25,000 patients. And this will be patient-driven, but there'll be more to come on that. Okay, so let's step away from the genetics and go into biology for the last few minutes and talk about how we actually can leverage this to an advantage. So as I mentioned, these 4 mutations are associated with the disease myelofibrosis progressing. One of them is a gene called IDH1 and 2. Dr. Mesa mentioned this earlier. And in fact, if you look at patients when their disease progresses to a more advanced form or leukemia, this is one of the most common mutations that we see, 3rd most common, 20 or 25% of patients. Whereas in patients who just have an MPN before transformation, it's maybe 3 to 5%. Now this gene, IDH, the protein is involved in metabolism. It's a normal part of metabolism. When it gets mutated, it actually produces a poison that poisons other metabolically inclined genes, and that actually causes disruption to the DNA, which can lead to leukemia, which is a fascinating story. So the question is, let's build models. Let's see, what does it really do, right? At least the patient data says to us it's associated with things going in the wrong direction. Can we validate that? And so this was done a couple of years ago, whereby JAK2 mutant mice that get polycythemia vera were engineered to also have this mutant IDH protein through a series of steps. And as it turns out, they do get a more advanced disease, exactly as we see in humans. So the only, So I'll come back to this slide in a minute. They do get a more advanced disease. Now, as it turns out, and this is, I think, an important breakthrough within the last 5 to 7 years, there is a drug called enasidenib, which was FDA approved for the treatment of leukemias in IDH2 mutant patients. This is a pill, and it was able to induce remissions in roughly 30 to 40% of patients, whereas chemotherapies traditionally, somebody who has leukemia that's not responding, may work 10 to 20% of the time. This is a breakthrough, and this is important. And this is now FDA approved fully for the treatment of patients with AML. So the question then to us was, well, fine, if you have patients, or in this case mice, that have a JAK2 mutation and have an IDH2 mutation, can you give them a JAK inhibitor and an IDH inhibitor, and does it do anything? Well, that was the question that was posed. These mutant mice were treated with nothing, with a JAK inhibitor, with the IDH inhibitor, or with both. What happens? Well, if you look at the sizes of their spleen, and again, I showed you what a leukemic spleen looks like in a mouse, you can appreciate that in red, if you combine both drugs, the spleen is much more markedly shrunken than with any of the two drugs by itself. And the same thing is true if you look at the white blood cell counts of the mice with both of the drugs. And going back here, if you look at the spleens of the mice that have the mutations, on the far right is the control. And all I want you to appreciate is that there's a lot of cells. All those black dots are cells. So you see that it is packed full of cells. And if you treat it with both drugs, you start to see clearing out, right? You start to see not so many of those round dots, all of which is an indication that this is actually doing something in the mice. So this is now translated fully into a clinical trial, right? This is a key example of taking the genetics, understanding the biology of the genetics, and then doing something about it. And this is the first genetically driven trial in MPN. This is a study for patients who have MPN that's a progressed to leukemia, or patients who have myelofibrosis with some increased blast, as long as they have an IDH2 mutation. And patients are being given Ruxolitinib and enasidenib in combination. So this is ongoing right now, and 6 patients so far have been enrolled. We've not seen any safety concerns in terms of combining the drugs. We need more data to find out whether this works well, but this is, to me, an important example of translating all of this genetics into something that's actionable, and that hopefully will make a difference. So I think with that I'll conclude and say that this is a complex space, right? And I know that when patients come in and say, this is what my genetic report says, it's perplexing, but it's perplexing not only because we know it may have implications, it's also because we don't yet have a complete understanding. This is what we need to advance and work on, but there is a lot of work, a lot of initiatives being done. Like I talked about, we have a progression project, which is really trying to understand what are the real implications here. And we have things like this, which are taking this and saying, fine, let's use that information to our advantage and intervene.